Okay, so imagine the entire genome is like this massive, unmapped continent. How in the world do you find your way around? How do you pinpoint one tiny, specific spot that's responsible for some really crucial trait? Well, that's where genetic markers come in. Think of them as the signposts, the landmarks, the GPS coordinates that let scientists navigate this incredibly complex map of life. So let's follow these signs and see exactly where they take us.
All right, here's our game plan. We're going to trace the story of these amazing tools, starting with the basics of what they even are. Then we'll look at the early days of observation, jump into the revolution that let us read DNA itself, and finally, check out the powerhouse techniques that are totally changing our world today.
So, first things first, what exactly is a genetic marker? Let's break it down. Let's start with the problem, because that always makes things clear. Imagine you're a scientist trying to create a plant that's resistant to a certain disease. You know there's a gene for that resistance, but you can't see it. It's invisible. So how do you follow that gene from one generation of plants to the next? You're going to need a landmark, something you can see that's always inherited right alongside it.
And that's our answer. A genetic marker is basically an experimental tag. It's a known feature. It could be a visible trait or just a specific snippet of DNA whose location on a chromosome is known. And because it's physically super close to our gene of interest, it gets passed down to the next generation right alongside it. It's like a bright flashing signpost that says, hey, the important gene you're looking for is right around here. Now, way back before we could read DNA directly, scientists had to get really clever. They used markers that they could literally just observe, either with their own eyes or with some pretty basic lab equipment. These were the classical markers. And these markers came in three main flavors. First, you had morphological markers. These are the most intuitive ones. Things you can see, like the color of a flower, or whether a seed is wrinkly or smooth. Then there were cytological markers, which involved actually looking at the chromosomes under a microscope to spot differences in their structure, like their banding patterns. And finally, biochemical markers, where scientists looked for subtle variations in proteins, like different forms of the same enzyme. You have to admit, this was incredibly resourceful. But these methods had some pretty big drawbacks. For one, there just weren't that many of these kinds of markers, not nearly enough to map the whole genome. And worse, something like plant height could be affected by the environment, like how much water it got, not just its genes. That made them pretty unreliable sometimes. It was clear scientists needed a much better tool.
And that better way? It arrived with a massive revolution. The ability to read the DNA code directly. This changed everything. It shifted the whole field from just observing the effects of genes to analyzing the genes themselves.
So, what would the perfect DNA marker look like? Well, here's the dream scorecard. You want it to be highly polymorphic, which just means there are tons of variations out there to find. You want it to be co-dominant, which is key. It lets you clearly see if an organism got a trait from just one parent or from both. And of course, in a perfect world, it's cheap, it's easy to use, it's reliable, and you can find these markers all over the genome.
Okay, so let's meet our first contender in the DNA era, RFLP. This was the pioneering technique that really, truly kicked off the age of DNA markers.
RFLP is a mouthful, I know, restriction fragment length polymorphism, but the idea behind it is brilliant. Imagine you have these tiny molecular scissors, these enzymes, that only cut DNA when they see a very specific sequence of code. Well, if there's a mutation, that code changes, and suddenly the scissors don't cut there anymore. This creates different-sized fragments of DNA, giving each individual a unique DNA barcode.
This diagram shows us how it worked. First, as you can see, you cut up the DNA with those molecular scissors. Then, you use a process called electrophoresis to separate those fragments by size. The small ones move faster than the big ones. And finally, you add a radioactive probe that lights up just the fragments you're interested in, revealing that unique barcode. It was a pretty involved process, but for the first time, it made these invisible differences in DNA totally visible.
And RFLP was a huge leap forward. It was co-dominant, it was very reproducible, but man, it was a beast to work with. It was slow, you needed a ton of really high-quality DNA, it was almost impossible to automate, and yeah, you were often dealing with radioactivity. So it was a breakthrough for sure, but the world was definitely ready for the next big thing.
And that next big thing was the polymerase chain reaction, or PCR. This technique wasn't just an improvement, it was a complete game changer. Suddenly you could take one tiny, almost invisible piece of DNA and make billions of copies of it. The need for big, perfect samples just vanished. This unlocked a whole new world of science, and it supercharged the development of a whole new family of markers that were faster, cheaper, and way more powerful.
This table really breaks down the new generation of PCR-based tools. First, you've got RAPD. Its genius was its simplicity. You could just amplify random bits of DNA without even knowing the sequence ahead of time. Super quick and easy to get started, but that randomness was also its biggest problem. It wasn't always reproducible. Then came SSRs, or microsatellites. These look for short, repeating sequences in the DNA, kind of like a genetic stutter. They are super variable, code-dominant, and really reliable, which made them the gold standard for a long time. The only catch? You needed to know some of the DNA sequence to design the test. And that brings us to the current champion, SNPs. These are tiny, single-letter changes in the DNA code. And their superpower is their sheer abundance. There are millions of them. This incredible density, plus the fact they're perfect for automation, makes them the absolute go-to tool for today's massive genetic studies.
This chart really puts that evolution into perspective. If you think about resolution power, the ability to see even the tiniest genetic differences, you can see the clear progression. RFLP was a good start. It gave us a medium resolution picture. SSRs were a big step up, much higher resolution. But SNPs, they give us the highest resolution of all. It's like going from a blurry photograph to a stunningly detailed, high-definition image of the genome.
So we've seen how the technology has evolved, from looking at flower petals to reading individual letters of code. But why does any of this actually matter? Let's talk about how these markers are being used right now out in the real world.
Well, the applications are just huge. Scientists use them to create incredibly detailed gene maps, pinpointing exactly where specific genes live on chromosomes. They're absolutely critical for diagnosing genetic diseases in both people and animals. In agriculture, a process called marker-assisted selection lets breeders develop better, stronger crops at lightning speed. And for historians of life, these markers let us trace the evolutionary story of species back through millennia.
And this really brings us to the main takeaway. There is no single best marker that works for everything. An SNP might be the perfect choice for a huge automated study, but an SSR could be way better for a smaller project looking at genetic diversity. The most important thing is for scientists to choose the right tool for the specific question they're trying to answer. It's all about picking the right signpost for the journey you're on.
You know, when you look back from just observing the color of a flower to being able to read single letters of DNA, the evolution of genetic markers is really a story of seeing things with more and more clarity. It just makes you wonder, as our tools get even sharper, what new secrets of the genome will we finally be able to track next?